It is a must that you have watched the video on alleles before this. If you have done so, we begin with Mendel. Gregor Mendel was born more than 200 years ago. He was a monk and he enjoyed growing peas. Many, many peas. One of his experiments involves crossing purebred yellow pea with purebred green pea. Purebred simply means they are homozygous for their colour. It turns out all the F1 generation has peas that are yellow in colour. F1 has nothing to do with car racing. The car wasn't invented until 2 years after Mendel had passed away, mind you. Just take it that the F stands for future generation and 1 is the first future generation. Since plants have both male and female parts, he made the offspring self-fertilise. In the end, the green phenotype appeared again in the second generation, or the F2 generation. Not only did he cross peas of different coloured seeds, but he also did so for the appearance of the seed, the colour of their flower, and the height of their stem. All the parental generation are purebred, and all the F1 generation are of the same colour, shape, and height. This is where he coined the term dominant phenotype, to label the phenotype seen in the F1 generation. Self-fertilization of the F1 generation caused the hidden phenotype to appear again, and he termed those as recessive. The dominant phenotype definitely appears more than the recessive phenotype in the F2 generation, and you can see the ratio is close to 3:1. To understand why this is so, we first have to touch on meiosis and the distribution of alleles. Meiosis is a cell division that produces sex gametes, which is a fancy way of saying sex cells. Remember your pair of homologous chromosomes with 2 pairs of sister chromatids. The pair consists of 1 chromosome from your mum and the other from your daddy. Let's take, for example, a pair of homologous chromosomes that are homozygous dominant for gene A, recessive for gene B, and heterozygous for gene C. At the end of meiosis, after the homologous chromosomes split, followed by the splitting of sister chromatids, you will be left with 4 sex cells. 100% of them will have the dominant A allele, and all 4 will have the recessive B allele. For the C allele, you can see that 50% of the gametes will have the dominant C allele, while the other half will have the recessive C allele. With the knowledge of how alleles are distributed and their percentages, we can explain Mendel's observations using a Punnett square. Let us use seed colour as an example. Capital Y for the yellow seed allele, and lowercase y for the recessive allele, which produces green seed. Draw the 2x2 table and we are ready to go. The purebred yellow pea will contribute 100% dominant Y alleles, while the green pea will contribute 100% recessive alleles. All of the F1 generation will inherit 1 dominant Y allele from the yellow pea parent, and the other allele inherited will be the recessive Y allele. As a result, all of the F1 generation will be heterozygous, and as all of them appear yellow, we determine that the yellow seed allele is dominant. For the second part of Mendel's experiment, he allowed the pea to self-fertilise, meaning it is a cross between heterozygous gametes. 50% of the gametes will have the dominant allele, while the other 50% will have the recessive allele. For the F2 generation, 50% of them inherit the dominant allele from the pollen grain and 50% inherit the recessive allele. The same goes for the alleles from the egg. 50% of the alleles passed are dominant and 50% are recessive. So for the F2 generation, the ratio of yellow to green seed will be 3:1, because the seeds with homozygous dominant and heterozygous genotypes will be yellow, while only 25% are homozygous recessive and will be green. Another possible combination is a cross between a heterozygous and a homozygous recessive. Let us take a look at the crossing of a heterozygous tall pea plant and a short pea plant. Tall is the dominant phenotype. Draw the 4 square and indicate the alleles on the outside. The tall parent will contribute 50% dominant alleles and 50% recessive alleles, while the short parent will only contribute the recessive allele. Half of the offspring will have one dominant allele, while the other half will receive the recessive allele. By adding another recessive allele to all the offspring, we will see that 50% of the F1 generation are heterozygous, while the other 50% are homozygous recessive. So the ratio of tall to short pea plants will be 1:1. Most of the time a Punnett square will be sufficient, especially for MCQ and combined science biology. However, for pure science students, you will need to know the genetic diagram. Genetic diagram is a fancier way of representing the crossing and mixing of alleles after fertilization. Let us use the crossing of a purebred pea plant with a purple flower with a purebred white flower. Purple is the dominant trait, so we will use a capital P for it and a lowercase p for the recessive allele. For a genetic diagram, you will have to list out similar items as in a Punnett square, namely the phenotypes of the parents, their genotypes and gametes, and the genotype and phenotype of the F1 generation. For genotype, we have 2 capital P for parents with purple flowers and a lowercase p for the white flower. Meiosis will take place and the alleles will separate. This big P can pair with this small p to produce a heterozygous offspring, or it can also pair with the other small p to also give a heterozygous plant. For the other big P, it is the same. It is only possible for it to pair with a small p, so all the F1 generation will be heterozygous, while all their phenotypes will be purple. For the F2 generation, it is just the self-fertilization of the F1 pea plants, which are all heterozygous, so all parents are purple, and their genotype is a capital P and a small p. Each will have 50% dominant alleles and 50% recessive alleles. The dominant P has an equal chance of matching with either a dominant allele or a recessive allele, and we will end up with a homozygous dominant genotype and a heterozygous one. Those recessive alleles also have an equal chance of matching with either a dominant allele or a recessive allele, which will give us a heterozygous and a homozygous recessive genotype. The F2 phenotype will be 75% purple and 25% white. Hence, the ratio of purple to white is 3:1. Besides using genetic crossing for alleles, we can also use it for chromosomes, as is the case with gender determination. Sex in humans is determined by our sex chromosomes, specifically whether we have a Y chromosome or not. For males, the genotype consists of one X and one Y chromosome, while for females, the genotype is double X. We can simply figure it out using a Punnett square, with the only difference being that we write the chromosomes instead of alleles. 50% of the sperm will have an X chromosome, while 50% will have a Y chromosome. For the eggs, all of them will only have the X chromosome. The result is identical to the previous cross we did, with a heterozygous and recessive phenotype. And the ratio of males to females is 1:1, which is the observation we see around us. Or is it? Is the Y the dominant chromosome? Yes. Is the X the recessive phenotype? Yes, as well. But this goes to show that the male is the sex responsible for the gender of the offspring. There are also situations where there are more than 2 alleles, and we term this as multiple alleles. We will also cover codominance. In the case of blood groups, instead of using a small or big letter to represent the alleles, we use I followed by a superscript of either A, B, or O. So please remember to use this format to represent alleles for blood groups. The O allele is recessive to the A and B alleles. This is because the A and B alleles produce their respective antigens on the surface of red blood cells. Because of the multiple alleles, this leads us to 6 possible genotypes. The corresponding phenotype is blood group O, when you have two O alleles. You will have blood group A if you have two A alleles or just one A allele paired with an O allele. There are also two possible genotypes for blood group B, which are either two B alleles or one B and one O allele. If a person has an A and a B allele, he or she will have AB blood group. AB blood group is an example of codominance. This is where two alleles controlling a trait, in this case the blood group, both express their phenotype, resulting in AB blood group, where the red blood cells have both A and B antigens. It must be noted that in cases where an intermediate trait is expressed, such as grey mice from white and black parents, it is known as incomplete dominance. Lastly, here are some possible crosses and their results. A parent with AB blood group and another with O will have children with either blood group A or B with equal chances. All the offspring of a parent that is homozygous for A and another that is homozygous for B will have AB blood group. Lastly, if someone who is heterozygous for blood group A and someone with AB blood group are to have children, the chance that the offspring will have blood group A is 50%, with the remaining 25% and 25% being the chances of having blood groups B and AB, respectively.